Hello. We are going to start today with Chapter 3, the cellular level of organization. So let's first look. Here's a cell. This is what we see if we get down to the microscopic level of what is actually makes up the inside of a cell. So we are going to break this apart and look at all the different parts that make up each individual cell. But first, what is a cell? Cells are the basic living structural and functional units of the body. When we study cells, it's known as cell biology or cytology. All cells have three main parts. They have a plasma membrane, they have cytoplasm, and then they have a nucleus. That plasma membrane is the outer surface of the cell. Our cytoplasm is what makes up inside the cell. So there's a cytosol, some fluid, liquid in there. There's different organelles that all have different functions, and different cells have different amounts of those organelles depending on where they're located in the body. Some of those organelles are a cytoskeleton. We have ribosomes, endoplasmic reticulum, a Golgi complex, lysosomes, peroxisomes, and mitochondria. Our nucleus. Our nucleus is where the DNA is kept. And within that nucleus, we have chromosomes. Our chromosomes is a single molecule of DNA associated with proteins that contain some hereditary units or genes. That plasma membrane is kind of a flexible border that surrounds the cell, and it also contains the cytoplasm of the cell. It kind of has this fluid mosaic model because it kind of moves around to fill in spaces. It kind of shrinks down or gets bigger depending on all the spaces that the cells may need to go. So within this fluid membrane, we have lipids that help make up this membrane, along with many different proteins. Some of the proteins are freely floating, so they're anchored in specific locations, while other proteins move around the membrane, and it allows molecules and ions to go in and out of that cell. Some are signal receptors that link the plasma membrane to the intracellular or extracellular proteins. This lipid bilayer, it's bi, so it's two. It's two layers back to back. And it's made up of three different types of lipids. We have phospholipids, so phosphate-containing lipids or phosphorus, cholesterol, and glycolipids. Glyco has glycogen. Most are phospholipids because they contain phosphorus. They have, or the lipid bilayer, that plasma membrane, is considered to be amphipathic. So it's got polar and nonpolar sides. If we look, we have a polar part that faces the outside. That polar part is hydrophilic. Philic means it likes water. So this lipid bilayer, the outside is hydrophilic, contains phosphate, and it likes water. The inside is two fatty acid chains or tails. They face each other. This nonpolar part is made of hydrocarbons and it's hydrophobic. It does not like water. So the two hydrophobic sides come together like, while the hydrophilic, the heads face the outside. Cholesterol is a weak amphipathic, and it is kind of more interspersed among other lipids on both sides of that membrane. The hydroxyl group on cholesterol, that's the only polar part of cholesterol. So that hydroxyl group actually forms hydrogen bonds with the polar heads of the phospholipids and the glycolipids. So we could get some cholesterol molecules kind of interspersed in here, and they've got hydrogen bonds holding them together. Glycolipids kind of only appear in the membrane layer that faces the extracellular matrix. So this is my outer layer. I have glycolipids that face out. They do not face in. We also have a lot of proteins in this membrane. Proteins can be integral or peripheral, and they help to move things across the membrane. So our integral proteins, those are firmly embedded because they have covalent bonds to those fatty acid chains, but they are considered amphipathic. So if I have my lipid bilayer here, I can have some integral proteins. They kind of extend in. So these integral proteins help bring things from one side of the membrane to the other. We also have a transmembrane. These transmembranes proteins, they extend the entire length of this membrane wall. They go both into the cytoplasm and the extracellular fluid. We also have peripheral proteins. They're not very firmly embedded in that membrane. They're attached to the polar heads. Or they could also be attached to the different integral proteins. We have glycoproteins. Those are proteins with carbohydrates. Glycogen. Glycogen is a carbohydrate. And many of those are integral proteins. Carbohydrates are kind of known as oligosaccharides, which means they have very few sugars. We also have a glycocalyx, which is a carbohydrate portion of that glycolipid and glycoprotein that form some kind of sugary coat for that plasma membrane. These membrane proteins that are attached have many different roles. A lot of them are ion channels. You can have some ion channel here. This channel can allow potassium to flow into and out of the cell. We have carriers. These carriers take ions or other molecules and take them from one side of the membrane to the other. We have receptors. Some kind of receptor. These receptors are sites that bind specific molecules. And what usually binds to those receptors is a ligand. Some kind of neurotransmitter through the nervous system and the muscular system. It could be some enzymes, it could be certain proteins. But what these ligands do is they open the channel, and then once the ligand detaches, it closes the channel back up. We have enzymes. As we talked about before, enzymes help to catalyze reactions so they do not require as much energy for that reaction to actually occur. And we use enzymes both inside and outside of the cell. So these enzymes are used along that plasma membrane to help with reactions. We have linkers. Linkers will actually attach one cell. Here's one cell membrane here. Here's another cell membrane here. Linkers. They attach the two cells to each other. It helps hold everybody in place. We have cell identity markers. These are specific receptors on cells that identify the cells as our own cells instead of some kind of foreign cell. That way our body knows, oh, we need to destroy these foreign cells. They do not belong here. Let's get rid of them. So here's some of our different channels. We have an ion channel here that is allowing ions just to freely flow. Some of them are specific and some of them are just general and let any ions through. Here's a carrier. It is taking these substrates or substances to be transported from one side of the membrane to the other. My ligand. This is binding. This helps to alter that function of that cell. It could open up a channel. It could trigger the cell to do something. We have substrates or enzymes. They bind to these proteins to catalyze some kind of reaction, then they release and go somewhere else. Our linkers, they help to bind one cell to the next cell, and then our cell identity markers. These help us identify our cells from foreign cells. This membrane is very fluid. It moves around very easily. So the membrane, the proteins within it, they all kind of rotate and move side to side in their bilayer. These proteins are always changing places, different places going around. It just depends on the double bonds that are created. And each of the double bonds actually puts a kink in that fatty acid. So here's our cell with our fatty acid chain. We have a kink here. This kink is a double bond. And what that does is it creates space between this cell and the cell next to it. That way things can easily move around. It has enough space for those membrane channels to move from place to place. It allows the membrane to move more fluidly. It also allows for self-sealing when injured. Like a great example is with infertility. They actually use what's called an intracytoplasmic sperm injection into the oocyte, which is the egg from the female, is injected with the sperm from the male. It's actually injected inside that oocyte. And then as the needle is removed, the membrane of that cell moves around and it self-seals back up. Our membrane, it's permeable to some things and it's impermeable to other things. It just depends on the size, the charge, the need. These all determine what goes through the membrane, what cannot pass through. So cells are technically selective permeability. So they allow substances to pass through while others will not pass, just like I said. So that lipid bilayer, easily let oxygen in, they let carbon dioxide in or out, and they let steroids in or out. They're somewhat permeable to small uncharged molecules like water. Water can get in and out somewhat easily. And urea. They are impermeable to ions in large uncharged molecules like glucose. So they need some kind of carrier or transmembrane protein that allows them to move from one side of the cell membrane to the other. When we're moving things from one side of ions, substances, substrates, different substances, when we're moving them from one side of the membrane to the other, they are moving along what's called a concentration gradient. So a concentration gradient is a difference in concentrations of the chemical from one place to another. Like one side could be more positive, one side could be more negative. The cells try to kind of even itself out. One side could have more water, one side could have less water. They try to even themselves out. So more ions and molecules are more concentrated in either the cytosol or the extracellular fluid. Oxygen and sodium ions are more concentrated in that extracellular fluid, while potassium is more concentrated inside the cell. So we have an electrochemical gradient that is created because of the differences in charges between the two regions. And that electrochemical gradient is actually known as the membrane potential, because that's the charge difference between the inside of the cell versus the outside of the cell. So the concentration gradient and the electrical gradient help to move substances across that plasma membrane. Substances tend to move down its concentration gradient. So an area of high concentration, it'll move down to an area of low concentration. Kind of like if you've ever put food coloring in water and how if you don't mix it up, you just let it sit, it eventually spreads to the entire glass. So we also have an electrochemical gradient, which is the concentration gradient and the electrical gradient on movement of a particular ion. So what are the chances of sodium moving into or out of the cell, or potassium moving in and out of the cell? Some things are done through an active process. Some are done through a passive process. When it's a passive process, it's usually simple diffusion is the best example for it, because substances move down their concentration gradient using kinetic energy, or as we said before, energy in motion. While the active process is when cells have to use their energy. They have to use the ATP that they're creating to move energy uphill against its concentration gradient. So that's when our active transport channels come in. That's where vesicles come in. Vesicles help transport things from one side of the membrane to the other, either into the cell or out of the cell. So let's look at diffusion. Diffusion is a random mixing of particles in a solution that occurs because of kinetic energy. So again, it's already energy in motion, so it does not require energy for diffusion to work. So solutes dissolve substances, and the solvents, the liquids doing the dissolving, undergo diffusion, kind of like that food coloring in water. So when you first put food coloring in the water, it's very concentrated. There's a lot of color right in that area, but it'll slowly diffuse to the rest of the container that it is put into. So there's different things that affect diffusion. How steep of a concentration gradient is there? The higher the concentration gradient, the higher the rate of diffusion. So everything wants to get evened out as fast as possible. Temperature. The higher the temperature, the faster diffusion is going to happen. The mass of the diffusing substance. If those particles are very large and heavy, they have a large mass, they're going to diffuse very slowly. We also have to look at the surface area of that membrane. If that surface area is a lot larger, that is going to cause diffusion to happen faster because there's a large area that needs to be covered. But that distance, the distance that particles need to move during diffusion, the greater that distance, the slower the diffusion again. Simple diffusion. Simple diffusion is the easiest kind. Nothing has to happen on any end to make it something diffuse. It's a very passive process. It happens freely as things travel through that plasma membrane. We don't use proteins. We don't use anything. They just go straight through that plasma membrane so that nonpolar hydrophobic molecules can move by simple diffusion, like oxygen, carbon dioxide, nitrogen gases that we also breathe in. Fatty acids go through. Fat-soluble vitamins, A, D, E, and K are the fat-soluble vitamins we have in our system. Small polar molecules can travel by simple diffusion. Water can. Urea and small alcohols move by simple diffusion. We use simple diffusion in our lungs. This is where oxygen and carbon dioxide need to be exchanged very quickly. So we have a very thin layer of cells for oxygen and carbon dioxide to get through. And because of this, simple diffusion happens very quickly within our lungs. So here's the example I was explaining. There's blue food coloring in this tube, and then there's water added. If we look, the blue food coloring starts out towards the bottom. It starts to move up towards the middle, and eventually the entire test tube is filled with blue water because that blue food coloring spread out. Facilitated diffusion is still technically diffusion because it doesn't require energy. But facilitated diffusion is where solutes, there could be too polar, too highly charged, so they don't move through that lipid bilayer through just simple diffusion. They contain integral proteins, and those integral membrane proteins actually assist substances, molecules, ions moving from one side of the membrane to the other. They could go through channels, or they can technically go through a carrier. Channel-mediated facilitated diffusion is when solutes move down that concentration gradient across that lipid bilayer through a channel membrane, a membrane channel. So that channel is going from one side, taking those ions from one side to the other. Potassium, we have specific channels for this. We have potassium channels. We have chloride channels. We have sodium channels or calcium channels. They're very specific to one type of ion. Some only let potassium in. They only let sodium in. They only let chloride in or out. Others just open their gate or close their gate depending on the chemical or electrical charges that are needed. Carrier-mediated diffusion is, again, this diffusion, so it's moving down its concentration gradient. We're not using ATP. So these solutes bind to a carrier on one side of the membrane, and they are released on the other side of the membrane after that carrier has changed its shape. Glucose, fructose, galactose, and some vitamins are very commonly use these carrier-mediated diffusion to get through. So we create ATP from glucose, from fructose, from galactose. It can all be done. And those membranes, they need to maintain that selective permeability, because as we've talked about with homeostasis, we need to keep that homeostasis in our body. Not just the whole body, but each individual cell has to maintain its homeostasis for cell life to work. So here's some of the different ways that things can travel across the membrane. We have our simple diffusion, where everything just goes through that plasma membrane from one side to the other. Channel-mediated facilitated diffusion, this ion or molecule is using a channel to get from one side to the other. While carrier-mediated facilitated diffusion, this compound is using a channel. It goes fit into the channel. The channel changes its shape, so it's released to the other side into the cytosol in the cell. We've looked at diffusion, so let's look at osmosis. Osmosis is technically diffusion, but it's where there's a net movement of solute through a selectively permeable membrane. So it's the movement of a solvent through a selectively permeable membrane. Again, it's a passive process. And the solvent is always going to be water from an area of higher concentration, and then it moves itself to an area of lower concentration, just like with diffusion. But osmosis uses that water because that water can pass through the plasma membrane different ways. It can move between phospholipid molecules by simple diffusion, or there are aquaporins, which are these integral proteins that are found in the plasma membrane, and they function as a water channel, letting waters in and out. These aquaporins can help to control the water content in that cell. This is how we produce cerebrospinal fluid, aqueous humor, tears, sweat, saliva, and the concentration of urine. It's all done through aquaporins. Mutations happen. When mutations happen, we get something that's can be called cataracts. That's when the aqueous humor in the front of our eye is not being drained out. It causes a lot of pressure problems. Diabetes insipidus does this. Our salivary glands can dysfunction. And we can also have neurodegenerative diseases. Osmosis only occurs when that membrane is permeable to water, but not permeable to some solutes. And because of this, it causes a hydrostatic pressure. And that's a pressure that is applied on any fluid that's in some kind of confined space. And we also have what is called osmotic pressure. It's when a solution with an impermeable solute is exerting a force. So let's look at this. So here we have a semi-permeable membrane or selectively permeable membrane here. We have water on one side, and then on the other side, we have water with some kind of solute. Water passes through the semi-permeable membrane. So because of the hydrostatic pressure, water actually travels from one side to the other. But if we apply some kind of osmotic pressure, which is what we do with this piece here, it levels everything out and restores it back to its starting condition. That helps cell volume remain constant by having that osmotic pressure. We don't want cells to get too much water in them or have too little water at the same time. So the tonicity is actually a measure of the solution's ability to change the volume of the cells by altering their water content. We want to know, are cells going to change their size based on the type of water? This is where we start looking at isotonic solutions, hypotonic, and hypertonic solutions. We try to maintain an isotonic environment in our body. So when we have an isotonic environment, cells maintain their normal shape, size, and volume. We keep our normal cell size. But with a hypotonic, the concentration of the solutes is lower than the cytosol. So this is opposite of what you think it would be, because a hypotonic is where water enters the cells, then leaves, causing that water enters the cells, then leaves, causing cells to swell and burst. So hypotonic water, more water enters the cell, too much water. In a hypertonic solution, we have a higher concentration of solutes than the cytosol of cells. So water moves out of the cell. Hypertonic, water moves out. Hypo, water moves in. This causes the cell to start to shrink. That cell shrinkage is known as crenation. So here's an example with red blood cells. Here is a typical red blood cell. In an isotonic solution, water moves in and out of the cell with no problem. The cell maintains its shape and size. Hypotonic is where water enters, too much water enters that cell. The cell starts to swell, and in a red blood cell's case, it'll undergo hemolysis, so it will burst. In a hypertonic, more water is leaving the cell than entering. This is where the red blood cell is going to start to shrink. It gets smaller and smaller. So here's a chart to help kind of look at the three different types of solution, where water moves, where solutes are higher or lower, what could happen to the cell due to the type of solution that it's in, and the net movement of water. Which way does it go? Let's look at active transport. The active transport is to move ions or solutes that need to cross the plasma membrane against their concentration gradient. So we need to move something in a direction that it's not likely to go. So since we're moving it against that concentration gradient, we need energy to make it happen. So when we get our energy, there's two different types of energy. We have primary active transport that requires the hydrolysis of ATP, or adenosine triphosphate. Then secondary active transport is where we have energy is stored in an ionic concentration gradient. So how the energy is used is determines if it's primary or secondary active transport. Our primary active transport, we get our energy from ATP. That ATP then changes the shape of that carrier protein to pump substances, ions, molecules across that plasma membrane against that concentration gradient. Our cells use about 40% of its energy on active transport. The most common active transport pump is our sodium-potassium pump. Sodium and potassium get moved in and out of the cell. And it utilizes ATPase, which is that enzyme that breaks down ATP. So all the cells have thousands of sodium-potassium pumps. And they help to maintain the sodium maintain that low sodium concentration in the cell by pumping them into that extracellular matrix against the potassium gradient. It's crucial for maintaining normal cell volume. If we get too much sodium in something, it starts to bloat, get bigger, it swells. Our secondary active transport uses stored energy that is found in a sodium or hydrogen concentration gradient. It's used to help move substances across its membrane against its own concentration gradient. So sodium and hydrogen or hydrogen gradient, it tends to be established by the primary active transport system. That secondary active transport system will indirectly use the energy obtained by the breakdown of ATP. So with secondary transport, we have two types of transporters that are used: a symporter and an antiporter. Symporters move substances in the same direction. So we could be moving sodium, and we could say be moving hydrogen in the same direction. An antiporter could move sodium in one direction while it moves potassium in the other direction. So we have several antiporters and symporters that are powered by that sodium gradient. So here's some examples of antiporters. This first one over here is allowing sodium to come in and calcium out, while the second antiporter is allowing sodium in and hydrogen out. Our sodium gradient is technically moving into the cell. So our symporters, they might move sodium and glucose into the cell, or sodium and amino acids into the cell. They move other stuff too. These are just some examples of what's moved in and out of the cell. We also have transport vesicles. As I've mentioned before, but we didn't talk about what they were. They're little spherical sacs that can move things from outside of the cell in or from inside of the cell out. They are used all over the body. There's two different ways vesicles work. They can work by endocytosis. Endo moves in. So it takes that vesicle, it's formed from the plasma membrane and moves into the cell, while exocytosis will move stuff out of the cell. Exo, out. Both endocytosis and exocytosis utilize ATP. So here is endocytosis. As we have particles here that need to go into that plasma membrane will come up and around, and it forms a vesicle here. The vesicle will eventually come in contact with a lysosome that helps break it apart, and it helps to release whatever it was carrying before it moves back. And we start with the next vesicle for endocytosis. Exocytosis, on the other hand, will take something and release it from inside our cell to outside the cell. So let's look at the cytoplasm of cells here. The cytoplasm is what's found inside the cell. Within that cytoplasm, we usually have cytosol, which is the fluid that is found, and then the different organelles, along with the nucleus. So let's look at the different stuff found in this cytosol. The cytosol is a fluid portion of the cytoplasm that surrounds the organelles. About 55% of the cell's total volume is cytosol, and about 75 to 90% of it is water. Within that water, we find ions, glucose, amino acids, proteins, lipids, ATP, and waste products. It's where a lot of chemical reactions occur, which help the cell actually survive. It's also the site where we find enzymes that breaks down that glucose or catalyzes glucose that makes ATP. We also have a cytoskeleton, which is made up of protein filaments that extend through that cytosol that helps to maintain that cell's shape. Those are made of microfilaments, intermediate filaments, and microtubules. A microfilament. A microfilament is the thinnest of the cytoskeleton. It is composed of actin and myosin, and it's actually most prevalent along the edge of the cell. It helps to move the cell, and it provides mechanical support. It could move the cell. It could be extending outside of the cell to move things around. Microfilaments are actually what is the part of the cell that contracts when a muscle contracts. We'll go into great detail about how that works when we get to muscles. That's where it's involved with cell division and cell locomotion, so cell moving. It provides a nice mechanical support for the cell, and it could be responsible for the basic strength and shape of the cell. It helps to anchor that cytoskeleton to the proteins that are in that membrane. The microfilaments have a lot of jobs. We have intermediate filaments as well. Those are thicker than the microfilaments, but they're thinner than the microtubules. That's why they're intermediate. They're in between. There's many different proteins that make these intermediate filaments. They're kind of found in parts of the cell that have a lot of stress, mechanical stress. Could be a lot of pressure, it could be pretty much anything. But they also help to stabilize the organelles, helps to stabilize the nucleus. That nucleus needs to be protected because that's where your DNA is. Our microtubules are the largest of that cytoskeleton. They're kind of hollow tubes, they're long, and they're made of tubulin. Assembly begins in an organelle called the centrosome. We will go over what that is, but they help give the cell its shape. It functions in movements of the organelles and of those secretory vesicles. It helps get those secretory vesicles where they need to go. Chromosomes during cell division, those microtubules help with that. Cell projections like cilia or flagella, those are what come up off the top of the cell, just like these. These are our microvilli. They extend up off of the cell. You can see in the electron micrograph over here that they're like little finger-like projections that come up. Those are made of microfilaments. we have our intermediate filaments. You can see they're a little bit bigger. They're kind of interspersed throughout the cell body here. And they're protecting, they're around that nucleus. We also have our microtubule. So you can see they're bigger, they're thicker, they're hollow tubes. They come out from the centrosome here, is where they're being created. And they're around again and protecting this nucleus. We have a lot of organelles in our cells, and they all have some kind of specifics. They have specialized structure and they have very specific functions. One organelle does not do every job in the cell. Each organelle has its own specific job. So they help with cellular growth, maintenance, and reproduction of these cells. Again, we have those centrosomes. As we mentioned before, that's the microtubule organizing center. It's near that nucleus, and it has two components to it. It has a pair of centrosols, which are 9 clusters of microtubular triplets arranged in a circular pattern. And then they have a pericentriolar matrix, and that surrounds the centrioles. And then there's hundreds of ring-shaped complexes of the protein peritubulin. Tubulin are organizing centers for growth of mitotic spindles for cell division. So if we look over here, we've got the 3 microtubules together forming that microtubule triplet. They are all formed around in a circle. And they're kind of arranged in that centrosome. And they come out of and help protect that nucleus. Now let's kind of look at cilia and flagella. Those are made of microtubules. They're kind of that main component of them. They are motile surface. Those cilia are more like finger-like short projections that come out. There's hundreds to thousands of them on top of each cell. So each cilia actually has 20 of those microtubules in it. And then there's that plasma membrane around it to help kind of give it its shape. They like to work in a beading or like pattern. They are found in your respiratory tract, in that trachea. They actually help move mucus away from the lungs. So they're constantly beating mucus up away from the lungs. Because if these did not work well, mucus would start to collect in your lungs. And it would cause breathing problems. It almost start to feel like you're drowning in mucus. Kind of like with cystic fibrosis. Cystic fibrosis, their mucus is so thick that cilia can't work. It cannot pump the mucus away from the lungs. We also have what is called a flagella. The best example of a flagella in humans is sperm, because it is usually one piece and it'll move the entire cell. It'll beat back and forth to project that cell moving forward. So here is what that flagellum looks like. You can see it up in the picture up here. It's made of doublet microtubules, and they come together, they beat side to side and help move, say, that sperm where it needs to go. We have ribosomes within all of our cells. Ribosomes is where proteins are made. They're named because they have a high count of ribonucleic acids in them. So each ribosome is about 50 proteins. We get a large unit and a small unit that come together to form ribosomes. So once ribosomes are created, some of them attach to that cell surface, others attach to the endoplasmic reticulum, and others are free or unattached to anything within the cell. So here's how we show we have that large subunit that attaches to that smaller subunit. That's how we get that complete functional ribosome. This ribosome has come off of the rough endoplasmic reticulum up here. So let's look at what is the endoplasmic reticulum? It's made up of a membrane of flattened sacs or tubules. It comes off of that nuclear envelope. That nuclear envelope is the outer layer of that nucleus. It's that membrane that's around it. And it kind of connects and projects through the cytoplasm. We have what I was mentioning before, the rough endoplasmic reticulum. That rough endoplasmic reticulum is actually continuous with that membrane of the nuclear membrane off that nucleus. It kind of folds and flattens back on itself. It contains many ribosomes, and it's kind of the site of protein synthesis. So some of those proteins actually get attached to carbohydrates to form glycoproteins, or others may attach to enzymes to form phospholipids. The rough endoplasmic reticulum produces secretory proteins, membrane proteins, and organelle proteins. We also have what is called a smooth endoplasmic reticulum. That smooth endoplasmic reticulum actually extends from the rough endoplasmic reticulum to form a network of membrane tubules. That smooth endoplasmic reticulum, it's smooth because it doesn't have protein or ribosomes. And since it does not have ribosomes, it does not make proteins. Instead, the smooth endoplasmic reticulum makes fatty acids and steroids. Steroids that are made could be estrogen or testosterone. In the liver, that smooth endoplasmic reticulum helps to release glucose into the bloodstream, or it could detoxify lipid-soluble drugs. So different places, they could have different roles, but they still kind of technically do the same kind of thing. Because like in the liver, the kidney, and the intestines, that smooth endoplasmic reticulum removes a phosphate group from glucose 6-phosphate, so we can let the glucose go free into the bloodstream. That glucose will then be picked up and converted to ATP. In muscles, calcium is stored in that smooth endoplasmic reticulum. We'll go into how all of that works when we get to the muscle chapter. So here is our rough endoplasmic reticulum. All these little blue dots on here, those are all ribosomes that are making proteins. Our smooth endoplasmic reticulum is actually connected to that rough endoplasmic reticulum. And as part of this whole protein process, we have what is called a Golgi complex. Our Golgi complex is where most of the proteins that are made by the rough endoplasmic reticulum are transported to, where they go to mature. They become what these proteins are destined to become. So the Golgi complex has an entry or a cis phase and an exit or a trans phase. That entry or cis phase, it faces that rough endoplasmic reticulum, and it tends to receive proteins. That's how proteins get from that Golgi complex, or no, the rough endoplasmic reticulum to that Golgi complex through vesicles. These vesicles release the proteins to the Golgi complex. The proteins travel through the Golgi complex and they mature. They turn into whatever type of protein the cell needs or the body needs. Once they get to the end of the Golgi complex, as they travel through it, they get to the exit or transphase. This phase is the plasma membrane. And again, these proteins are enclosed into secretory vesicles again. And they either exit the cell or they're used within the cell. We also have intermediate saccules. So between the entry and the exit phases, they can add carbohydrates or lipids. This is where we form glycolipids or glycoproteins. Proteins then mature as they pass through that Golgi complex. So here's the whole process. We have our rough endoplasmic reticulum over here. We have all these little proteins. They go into a transport vesicle. They enter the Golgi complex. As they travel through the Golgi complex, they mature and they turn into whatever job they're supposed to do. They get put into different kinds of vesicles. They could be put into a transfer vesicle. They could be put into a secretory vesicle, a membrane vesicle, or a transport vesicle. Different proteins go to different places depending on where they are needed and what they mature into. Now our lysosomes. Our lysosomes are membrane enclosed vesicles that are formed from that Golgi complex. They contain different digestive and hydrolytic enzymes. They are used to break down some molecules, and they will digest worn down cells. They recycle cell parts, cell structures to be used again. The inside of lysosomes actually have a pH of 5. That pH of 5 is what helps them digest cell parts. Through the process of autolysis, they can also destroy the entire cell. And that helps to do either the cell is dying, the cell is worn out. The lysosomes will trigger autolysis. Sperm is a great example because they release some lysosomal enzymes that help to penetrate that oocyte, because whatever sperm gets there first is the one that gets to penetrate that oocyte and produce a zygote. So here is a lysosome. It's got many different digestive enzymes in it to be used for whatever process. It could be digesting cell parts. It could be digesting that entire cell. It's just dependent on what is needed. We have peroxisomes. They are called microbodies, and they contain oxidase because they remove hydrogen atoms. We have amino acids and fatty acids that are oxidized in peroxisomes as part of our normal metabolism. So that they oxidize toxic substances such as alcohol. So they're very abundant in the liver, because if we drink a lot of alcohol, that liver needs to detoxify our body. And those peroxisomes help make it happen. We also, if we take another damaging substances, our liver needs to detoxify it. Our liver naturally detoxifies our system. We also it's also created as a byproduct of the oxidation of hydrogen peroxide and other free radicals. We make superoxide. So our cells make superoxide, free radicals sometimes, just by breaking something else down. They contain catalase that breaks down hydrogen peroxide And they also have those enzymes. Once it creates that superoxide, it needs to destroy it right away. So those peroxisomes, as it creates that superoxide, breaking down hydrogen peroxide or something else, once that superoxide is created, it breaks it down. We don't want it in our system. Peroxisomes also can self-replicate. So if we need more peroxisomes, more start to replicate and create themselves. We have proteasomes. These proteasomes destroy unneeded, damaged, or faulty proteins. So if we create more proteins than we need for a certain task, proteasomes destroy them so we can use the pieces again. If we have a damaged protein, that's not good. Damaged or faulty are not good. They can create problems down the road as they get replicated. So we need to destroy them before they cause problems. So proteasomes kind of destroy those faulty or damaged proteins. They contain proteases, which are the proteins that cut the enzymes that cut proteins into smaller peptides so that they can be reused for those new proteins. They reuse the pieces when they break them apart. Our mitochondria. Our mitochondria is our energy creation source. They generate all our ATP for the cells. They tend to use aerobic respiration. Aerobic means it requires energy or oxygen to create the energy. They're very active in muscle cells, kidney cells, liver cells. They have lots of mitochondria. They need lots of energy to function. They also participate, that mitochondria, in apoptosis, which is a genetically programmed cell death. So if we get a lot of free radicals into our cells, DNA is damaged, growth factors are deprived, or the cells lack enough oxygen and nutrients. This triggers the mitochondria to release chemicals that triggers the cell to kill itself. So mitochondria can also self-replicate in times of great energy demand. Or they also tend to replicate right before cell division too. So mitochondria actually has their own DNA that are 37 genes long. The DNA that comes from the mitochondria only comes from your mother. There is no part of the mitochondrial DNA that comes from your father. So here is a mitochondria. As we can see, because they enlarged, we have a lot of ribosomes creating proteins. We have a matrix. We create all of our ATP in this organelle. This provides us with the energy our cells need to work. We have a nucleus. Not all cells. Most cells have a nucleus. Red blood cells, once they have matured and entered the bloodstream, they no longer have a nucleus. They start with a nucleus, and as they mature, the nucleus leaves the cell. Skeletal muscle, on the other hand, have a few nuclei. The nucleus is separated from the rest of the cell by what we had talked about before, a nuclear envelope. This nuclear envelope helps to separate what's inside the nucleus from outside. Again, the nuclear envelope consists of a nuclear or a lipid bilayer. And it's actually continuous with that rough endoplasmic reticulum. We also have nuclear pores on that nuclear envelope that allows substances to travel, water to travel back and forth, allows some ions, proteins to travel back and forth. We also, in that nucleus, we have a nucleoli. That's where our ribosomes actually get produced, is in that nucleoli. So it consists of a cluster of proteins, DNA, and RNA. It's where our ribosomal RNA is actually made. And it also assembles that ribosomal RNA into the ribosomal subunits that are used by our cell. We have a lot of nucleoli in muscle and liver cells because they need a lot of ribosomes to perform their jobs correctly. The nucleoli, as the cell is getting ready to divide through cell division, that nucleoli actually disappears. And then it forms again when the new cells are formed. We have chromatin in the nucleoli, which are the proteins, DNA, and RNA. And we have what is a genome, which is your genetic code for that entire cell. Every cell has the genetic code to make up who you are. So here is our whole nucleus with our nucleolus. We have again our lipid bilayer here. We have our nucleolus in the middle that houses our DNA and RNA. We have all these little nuclear pores. We have ribosomes that are attached. These all make up our nucleus and the nucleolus inside. Let's look at protein synthesis, because this is a process that helps to make our RNA and DNA. So proteins are determined by the physical and chemical characteristics of the cell. We have proteome, which are all of the organism's proteins. And then we have what is called gene expression. It's when a specific gene's DNA is used as a template to make specific proteins. And this happens through transcription, is where that information is encoded in a specific region of that DNA. So it is transcribed or copied to produce RNA. DNA is 2 strands, if we remember. RNA is 1 strand. So if we take that DNA, we transcribe one side of it, that would make an RNA strand. And then through translation, that RNA attaches to some ribosome, and that information is copied and it's translated into a corresponding sequence of amino acids to form new protein molecules. So RNA and DNA are both, they store our genetic information. And they store them in sets of 3 nucleotides together. So we have nucleotide 1, 2, 3. These are known as base triplets because they are transcribed together. Then we have another set, that's another base triplet. And we have what is known as a codon that travels along from one base triplet to the next base triplet and transcribes. So it matches up these amino acids with their matching pairs. Then it moves to the next set and does the exact same thing. And a given codon specifies a particular amino acid. So like I said, they match the amino acids with the specific base triplets. Our genetic code is the set of rules that regulate or relates to these base triplet sequences. And they respond to those codons. So our genetic code is telling us, okay, we have these 3 bases. these 3 amino acids need to attach. That's how they're going to work. In transcription, we have 3 types of RNA that are made from that DNA template. Messenger RNA, which directs the synthesis of proteins, tells us how to make different proteins. Ribosomal RNA makes ribosomes. And transfer RNA, these bind amino acids and holds them until incorporated into protein during protein synthesis. The proteins are made of many different amino acids linked together. RNA polymerase is what actually transcribes that DNA. And then at the end, we have what is called a terminator that signals the end of that DNA strand. So we're at the end of transcription. We're done with this set of DNA. So here's this whole process. We have our DNA. We have a codon, which is these 3 amino acids. They're also known, they're in base triplets. This RNA polymerase is transcribing, moving down the strand of DNA to make the specific part that it needs to transcribe certain proteins for the body, for whatever it may be needed. During translation, those nucleotide sequences in a messenger RNA molecule specifies the amino acid sequence of those proteins. So ribosomes carry out translation, and it has binding sites for that messenger RNA. Let's look at cell division. How do cells divide? So we know how DNA divides, how DNA creates new strands, RNA creates new strands. Those RNA strands are used to make proteins. Let's look at how cells divide. So most cells in the body can replicate. Most, not all. We have somatic cells, which is almost any cell in the body except for those germ cells. And a germ cell is a gamete, like the sperm or the oocyte, or any cell that is destined to become a gamete. You'll learn more about those when we get to the reproductive system and anatomy too. Cell division can happen through mitosis, and this is how somatic cells reproduce, or reproductive cell division. So those sperm and oocytes actually go through meiosis. So somatic cell division is when we duplicate everything inside of one cell and we form 2 cells with it. Each human cell has 23 pairs of chromosomes in there. And that is what's inherited from our parents. Our mom gives us one half, our dad gives us the other part of them. Homologous chromosomes are 2 chromosomes that make up each pair. So they're very similar. But then we have our sex chromosomes. Females have that XX chromosome and males have that XY. Y is what designates males versus females. And that during the interphase portion of somatic cell division, we replicate our DNA. We produce any additional organelles and cytoplasmic components that we need for cell division. This is very high metabolic activity. This is where most of the cells grow. It usually takes about 8 hours to complete. The mitotic phase actually has 4 phases in there. It has prophase, metaphase, anaphase, and telophase. During prophase, those chromatin fibers, they shorten, they condense and shorten into chromosomes. During that metaphase, those microtubules of that mitotic spindle we talked about, they align together And those centrosomes form chromatid pairs. We get an anaphase, is where those centromeres split and they start separating into two separate members, and they move to opposite sides of the cell. Then telophase is after chromosomal movement stops. Those two sets of chromosomes are at opposite poles, so the nuclear envelope or that plasma membrane comes together and they form two completely separate cells. So let's look at this. During interphase, phase 1, things start to replicate. We get more than one. We get two sets of everything. Prophase, things are separating even more. They're starting, that nucleolus disappears. Everything kind of moves and lines up in the center, and then they split into opposite pairs during that anaphase. Telophase, we are starting to get two separate cells. And by the end of telophase, we have two completely identical cells. Cells, as they replicate, they have three different destinies. Some cells, once they mature, they do not replicate. They just remain alive and they function. Other cells, their main job is just to keep replicating and growing. Those are usually some kind of stem cell. Cells also die as we went through apoptosis, programmed cell death, where a cell kills itself because there's something wrong with it, or it's just too old and not functioning well anymore. Let's look at meiosis. This is where those gamete cells reproduce. This is how we get new sperm cells, new oocytes. Meiosis actually occurs in two stages. We get meiosis 1 and meiosis 2. Meiosis 1 is where begins when chromosomal replication is complete. So we get prophase 1, where those chromosomes shorten and thicken, the nuclear envelope and the nucleoli disappear, and we get mitotic spindles. Metaphase 2 is where those chromosomes line up along that metaphase plate of the cells and with chromosomes side by side. Anaphase 1, those chromosomes start to separate and they're pulled to opposite poles of those microtubules. Then our telophase 1 is similar to telophase of meiosis. Meiosis 2 has 4 phases as well, but it's prophase 2, metaphase 2, anaphase 2, and telophase 2. They are all similar. They're similar to the same stages of mitosis. So what happens in meiosis is we go from 1 cell to 4 cells. That's where meiosis is different. We go from 1 cell to 4 cell versus mitosis, we go from 1 cell to 2 cells. As we get older, it's a normal process that happens with our body, but we find it hard, our cells find it harder to respond and adapt to homeostasis changes. We don't respond as well. On the ends of our DNA, we actually have what are called telomeres. These telomeres are specific DNA sequences, but they're only found at the tips of each of those chromosomes, and they help protect the tips from breaking, from erosion. But during each cell cycle, those telomeres get shorter, and then eventually they're gone. And this is what helps contribute to aging. This is what contributes to cell death. If you've ever seen anybody who's under a lot of stress, has very significant high stress, their telomeres are a lot shorter. So why people, when they undergo significant amounts of stress, age very quickly. other things that happen when we age. We could start to have uncontrollable or abnormal cell growth, cell division. This is where excess tissues form and they turn into some kind of tumor. We could grow a malignant tumor. That's a cancerous neoplasm. Malignant tumors are actually malignant because they undergo metastasis and they spread from one part of the body to another part of the body. We also have benign tumors. They do not metastasize. So unless they're causing problems, they can be surgically removed. If they're disrupting your normal body function, or they become disfiguring, they cause some kind of pain, because the only time they cause pain is if they're pushing on nerves or closing off blood vessels. The main kinds of cancer is we have carcinomas, sarcomas, leukemias, and lymphomas. A carcinoma involves epithelial cells. We'll go over epithelial cells in our next chapter, exactly what those are. But think of your skin. But epithelial cells are found throughout your entire body. We have sarcomas, which is cancer that actually comes from muscle fibers. Leukemias, they affect white blood cells. And lymphomas, lymphatic lymph nodes are affected. So as cancer tumors keep growing, we have the process of what's called angiogenesis. This is where blood vessels start to grow and they come in contact with these tumors, and they go through these tumors and provide these tumors with the nutrients that they need for these cells to keep replicating. As these new blood vessels are formed, this is how malignant cells can get from one part of the body to the next. A cell could come off, get into the bloodstream, and travel to some other part of the body. They can get off into the bloodstream, into the lymphatic system, travel somewhere else in the body. Because we know what cells look like for different parts of the body, we're actually able, when they look at tumors, determine, is this a primary tumor? Are these cells from where they pulled them from originally? Or are these cells from somewhere else in the body? Did they travel to get here? And that's what makes them malignant. So as these tumors keep growing, normal tissue around it starts to die because they're not getting the nutrients they need. This tumor is instead. When somebody experiences pain from tumors, from a malignant tumor specifically, it's because the tumor is pressing on nerve fibers. It's blocking the passageway for lymphatic tissue, for blood, for veins. And the problem that these malignant tumors have is we actually have anti-tumor defenses in our body, but malignant tumors are resistant to them. And that's why malignant tumors grow so well in our body, is they can resist any of the processes that we have to kill off malignant tumors. So there's different things that cause cancer in our body. They could be the air we breathe. That's why we talked about before in chapter 1, breathing clean air. If we're constantly breathing polluted air, if we need to drink clean water. The food we eat. This is why eating healthy foods that do not contain some kind of carcinogen, because these carcinogens can cause the mutations or permanent changes to our DNA that can cause cancer to grow. Oncogenes is when a portion of our genes are actually activated that shouldn't be. So it transforms a normal cell by activating something that shouldn't be activated into a cancerous cell. There's also oncogenic viruses. Some cancers actually have a viral origin. HPV is a great example. HPV is actually a virus that causes cervical cancer in women. And viruses are actually RNA or DNA. They get into cells, they infect cells, and they use all of our cells, organisms, or our organelles to replicate themselves. Then they release out and they go to the next cell, use those cells, organelles, use everything they need. They use up everything to replicate as much as they can. Then they destroy the cell when they're released out. And if they're going into all these cells into the women's cervix, it can trigger the formation of cancerous cells that forms a tumor that can cause cervical cancer. So here is a picture of cancer cells actually dividing in the body. Can show how they're different kinds of cancer cells as they divide in each part of the body. That is it for chapter 3 and the cellular organization of the body. Thank you. Bye.